Let's begin with another session for today entitled as Liposomes, a novel drug delivery system. We have an eminent speaker for today's session, Ms. Trupti Jadhav ma'am. Ms. Trupti Jadhav ma'am has been working as an assistant professor in Department of Pharmaceutics at SJIPR. She has completed her B Pharm from Shivaji University and her M Pharm from University of Mumbai. She has 4.5 years of industrial experience in corporate quality assurance department. She has 2 years of academic experience. She is a life member of professional bodies like a Maharashtra State Pharmacy Council and the Indian Pharmaceutical Association. With this brief introduction, I request ma'am to take over the session.
Good afternoon, one and all. Good afternoon, one and all. Thank you, Samiksha, for a nice introduction. So, dear students, for today's SCIENTIA session, my topic is liposomes, a novel drug delivery system. So, let's start with the presentation. So, this is the outline of my presentation. So, in which we are going to see what are liposomes, basic liposome structure, structural components of liposome, advantages of liposomes, classification, then methods of preparation of these liposomes, and evaluation, and lastly, the applications of these liposomes. So, now first we will start with the what are liposomes. Now, basically, these liposomes are concentrate bilayered vesicles in which an aqueous core is entirely enclosed by a membranous lipid bilayer. Mainly, this is composed of natural or synthetic phospholipids. Okay, so the size of this liposome ranges from some 20 nanometer up to several micrometer. So this is the basic liposome ka structure. Next, the lipid molecules are usually phospholipids. These phospholipids are amphipathic moieties with a hydrophilic head group and two hydrophobic tails. So, on addition of excess water, such lipid moieties spontaneously originate to give the most thermodynamically stable conformation. That is nothing but the liposomes, in which these polar head groups face outwards into the aqueous medium. So these polar head groups are face outwards into the aqueous medium, and the lipid chains turn inwards to avoid the water phase, which gives rise to double layer or bilayer lamellar structure. That is nothing but the structure of liposome. So this is the basic liposomal structure. In this, you can see this is hydrophilic region. This is hydrophobic region. If you want to incorporate your hydrophilic drug, then you have to incorporate, or that drug will get incorporated in this hydrophilic region. If any hydrophobic drug is there, then that hydrophobic drug will get incorporated in this hydrophobic region. Now, this lamella is the structural unit of our liposome. So, a liposome is a flat plate-like structure which appears during the formation of liposomes. So, the phospholipids bilayer first exist as a lamellae before getting converted into the spheres. So several lamella of phospholipids bilayer are stacked one upon the other during formation of these liposomes to form a multilamellar structure. Now, what are the structural components of liposomes? So mainly there are two components of liposome. First is phospholipid and second is cholesterol. Now, this phospholipids, these are the major structural components of biological membrane, such as the cell membrane. So, where two types of phospholipid exist. First is phosphoglycerides and second is sphingolipids, together with their corresponding hydrolysis product. Now, this phosphatidylcholine is an amphipathic molecule in which a glycerol bridge links a pair. This glycerol bridge links a pair of hydrophobic acyl hydrocarbon chain with a hydrophilic polar head group, that is a phosphocholine. This phosphatidylcholine is also known as lecithin, and that can be derived from natural as well as from synthetic sources. So this is the general phospholipid structure, which includes polar group. This is phosphate group, glycerol ka bridge between fatty acid chain. So this is hydrophilic head and hydrophobic tail. Next structural component of liposome is cholesterol. So this cholesterol by itself does not form any bilayer structure, but it can be incorporated into phospholipid membranes in a very high concentration, up to 1:1 or even 2:1 molar ratios of cholesterol to PC ratio, that is phosphatidylcholine ratio. This cholesterol acts as a fluidity buffer. After incorporation with this phospholipid molecules, it alters the freedom of all the motion of carbon molecules which are there in the acyl chain. So it restricts the transformation of trans to washy conformation. So this cholesterol incorporation increases the separation between choline head groups, and it eliminates the normal electrostatic and hydrogen bonding interaction. Now we will see the advantages of liposomes. These liposomes provide selective passive targeting to tumor tissues, increased efficacy and therapeutic index. Next, increased stability of encapsulated drugs, reduction in toxicity of the encapsulated agent. It also provides us the site avoidance effect, improved pharmacokinetic effects, then flexibility to couple with the site-specific ligands to achieve all the active targeting. Now, there are some disadvantages also of these liposomes. First disadvantage is physical and chemical stability, very high production cost. Therefore, we cannot go for this liposome. Drug leakage, entrapment, drug fusion, sterilization, rate of release and altered biodistribution, low therapeutic index and dose effectiveness. So these are the disadvantages. Now we will move towards the classification of these liposomes. So this classification of liposome is based on structural parameters. So multilamellar vesicles, oligolamellar vesicles, unilamellar vesicles, small unilamellar vesicles. So these are the classification of liposomes which are based on their structural parameters. On the basis of liposome preparation also, these liposomes are classified. So some vesicle prepared by reverse phase evaporation method, stable plurilamellar vesicle, frozen and thawed. So these are the methods of preparation by which we can also classify the liposome. Next, method of liposome preparation. In this, three method of liposome preparation are there. First is physical dispersion, second is solvent dispersion, third is detergent solubilization. In physical dispersion method, again 4 methods are there: handshaking, non-shaking, freeze drying and proliposomes. If you want to reduce the liposomes size, then there are again 4 methods like microemulsification, membrane extrusion, ultrasonication and French pressure set. If you want to increase the liposomal size, then there are again 3 methods: dried reconstituted vesicle, that is DRV, freeze thawing and induction of vesiculation by pH change. In solvent dispersion method, again there are 3 methods: ethanol injection, ether injection, water organic phase. In water organic phase, again it is divided into 3: double emulsion method, reverse phase evaporation and stable plurilamellar vesicles. And lastly, the detergent solubilization. Now, First, we will start with the physical dispersion method. In that, first method is handshaken MLVs. This is the simplest and most widely used method of physical dispersion. In this method, the lipid mixture of different phospholipids and charged components in chloroform is to methanol solvent mixture, that is 2:1 volume by volume ka ratio is taken. It is prepared first and then it is introduced into 250 ml round bottom flask, that is RBF, with round glass neck. In this flask is again attached to the rotary evaporator and it is rotated at 60 rpm. So the organic solvents are evaporated at about 30 degrees Celsius or about the transition temperature of the lipids used. Now, what is transition temperature? Transition temperature is nothing but the temperature at which the phases changes of that lipid. So the rotation is continued for 15 minutes and after the nitrogen is introduced into the evaporator and the pressure at the cylinder. Then this head is gradually raised until there is no pressure difference between inside and outside of the flask. So the flask is removed from the evaporator and fixed onto the manifold of lyophilizer to remove all the residual solvents. Next step is hydration of these lipids. After releasing the vacuum and removal from the lyophilizer, this flask is flushed with the nitrogen. So then 5 ml of saline phosphate buffer is introduced. The flask is attached to the evaporator again and it is rotated at room temperature and pressure at the same speed or below the 60 rpm. The flask is left rotating for at least 30 minutes or until all the lipid has been removed from the wall of the flask and has given a homogeneous, and it gives a homogeneous milky suspension, milky white dispersion, that is nothing but the liposomes. Then we have to keep that suspension or dispersion at least stand for 2 hours to get our MLVs. Next, non-shaking vesicles. In this method, large unilamular vesicles can be formed with higher entrapment volume, or you can say a higher entrapment efficiency. They can be they used the same method except more care over the swelling procedure. So the hydration and swelling, these are the two processes which are involved in this method of preparation. So hydration is first of all initiated by exposing all the dried film to a stream of water saturated with a water saturated nitrogen for 15 minutes, followed by swelling in aqueous medium without shaking. In this method, solution of lipid in chloroform is to methanol ratio or mixture is sprayed over that flat bottom conical flask, and then the solution is again evaporated at room temperature by the flow of nitrogen through the flask without disturbing that solution. After drying, this water saturated nitrogen is passed through the flask until the opacity of the dried lipid film disappears. After hydration, this lipid is swelled by addition of some bulk fluid. So the flask is inclined to one side. 10 to 20 ml of 0.2 molar sucrose is added, or sucrose in distilled water is introduced down the side of the flask. Then the flask is slowly returned to the upright orientation. So the fluid is allowed to run gently over the lipid layer on the bottom of the flask. So the flask is again then flushed with the nitrogen and allowed to stand for 2 hours at room temperature or 37 degrees Celsius. After swelling, or the vesicles are harvested by swirling the content of the flask gently to yield a milky dispersion or suspension. So the suspension is again centrifuged at 12,000 rpm. From that we will get the large unilamular vesicles. Proliposomes. So the lipid, so in this method, lipid solution in chloroform is prepared and sorbitol powder is introduced into the 100 ml flask. The flask is then fitted into the evaporator and rotated slowly so that the powder tumbles gently off the walls to ensure all the good mixing and the solvent is evaporated in this case. The flask is again lowered into water bath at 50 to 50 degrees Celsius, 50 to 55 degrees Celsius. When a good vacuum has developed, an aliquot of 5 ml of liquid solution is introduced into the flask via the solvent inlet tube. So the solvent is absorbed completely by the powder, and the temperature of the bed is monitored. As evaporation proceeds, the temperature will get decreased. So a second aliquot is introduced slowly when the temperature begins to rise again. The temperature is allowed to rise to 30 degrees Celsius, and the vacuum is released, and the drying process is completed by connecting the flask, connecting the flask which is containing the powder to the lyophilizer, and leaving it to evacuate it overnight at room temperature. From that we will get the multilamellar vesicles. Next is freeze drying method. So this is another method of dispersing the lipid in a finely divided form. So prior to addition of this aqueous media, first of all we have to dry the lipid which is dissolved in suitable organic solvent. Then in this tertiary butanol organic solvent is used, and it is the most considered suitable solvent. After obtaining the dry lipid, water or saline can be added or introduced with the rapid mixing above the phase transition temperature, which gives you MLV, that is multilamellar vesicles. Now, to reduce liposome size, we can use this method, microemulsification liposome. This microemulsification liposome is prepared by the microfluidizer, which pumps fluid at very high pressure through a 5-micron orifice. Then it is forced along defined microchannels which direct two streams of fluid to collide together at right angles at very high velocity. After a single pass, the size will get reduced to 0.1 and 0.2 micron in diameter. So this is the microemulsification liposome. Next is sonicated unilamellar vesicles. In this, we have to take MLV in test tube. After that, MLV which is there in test tube, we are going to sonicate it for 5 to 10 minutes, which above the phase transition temperature. After that, we are going to centrifuge at 1 lakh RPM for at least 30 minutes at 20 degrees Celsius. Then we are going to decant all the top layer to get the sonicated unilamellar vesicles. Now, in this method, you can use bath sonicator as well as probe sonicator. Next method is French pressure cell. So this French pressure cell is invented by Charles Stacy French. In this technique, the large vesicles are converted to small vesicles under a very high pressure. So this technique yields uni as well as oligolamellar liposomes of intermediate size, and these liposomes are more stable as compared to the sonicated liposomes. Membrane extrusion liposomes. In this technique, vesicle contents are exchanged with the dispersion medium during breaking and resealing of phosphate lipid bilayer as they pass through the polycarbonate membrane. In this, less pressure is required as compared to French pressure cell, and it is used to process all the MLVs and LUVs. So two types of membrane, one is torches and another is nucleation trap. Now, next method, to increase size of liposome, freeze-thaw sonication method can be used. In this method, the entrapped material is again introduced into liposome after they have been formed. In this method, the freezing and thawing process is used, which ruptures and refuse SUVs, during which a solute equilibrates between inside as well as outside, and the liposome themselves fuse with each other, and it will get and they will increase their size. Next, dried reconstituted vesicles. So in this method, water soluble materials which we have to entrap are added to the suspension of empty SUVs, and they are freeze dried together. So the material for inclusion is present in the dried precursor lipid before the final step of addition of aqueous medium. These liposomes obtained by these methods are usually uni or oligolamellar vesicles. Next method is pH induced vesiculation. In this method, dry film of lipid is obtained in the round bottom flask using rotary evaporator, and last traces of all these solvent are removed using freeze dryer method. Then the film is hydrated with minimum quantity of water by hand shaking at room temperature. At this stage, materials to be entrapped inside the vesicle may be added in the water before addition to the lipid. Sodium hydroxide solution is sorry sodium hydroxide solution is uh, in, uh, added rapidly with the mixing into this suspension, and then the pH is reduced by addition of 0.1 molar HCl. Therefore, that pH will moves down to the 7.5, and because of which we will get the SUVs. Now, in this method, change in pH brings about an increase in the surface charge density of lipid bilayer, and this induces spontaneous vesiculation. So next is evaluation of liposomes. The liposomes, which are prepared by various techniques, are to be evaluated for their physical properties. Has this influenced the behavior of liposomes in vivo? So first evaluation parameter is particle size and particle size distribution. So both the particle size and particle size distribution of liposomes influence their physical stability. So these parameters can be determined by laser light scattering or transmission electron microscope. Now next is surface charge. This is the second evaluation parameter. In this, the positive, negative or neutral charge on the surface of the liposome is due to the composition of the head groups. So the surface charge of liposomes govern the kinetic and extent of distribution in vivo, as well as interaction with the target cell. So the method involved in the measurement of this surface charge is based upon the free flow electrophoresis of MNVs. It also utilizes a cellulose acetate plate dipped in sodium borate buffer of pH 8.8. About 5 moles of lipid samples are applied onto the plate, which is then subjected to the electrophoresis at 4 degrees Celsius for 30 minutes. The liposomes get bifurcated depending upon their surface charge, and this technique can be used for determining all the heterogeneity of charges in the liposome suspension, as well as to detect any impurities such as fatty acids. Next evaluation is percent drug encapsulated. So this is the most important evaluation parameter in case of liposomes. Quantity of drug entrapped in the liposomes helps to estimate the behavior of the drug in the biological system. Liposomes, these are the mixture of encapsulated as well as uncapsulated drug fractions. So the percent of drug encapsulation is done by first separating the free drug fraction from encapsulated drug fraction, and then the encapsulated fraction is then made to leak off the liposome into aqueous solution by using some suitable detergents. So there are the methods used to separate the free drug from the sample. First is mini column centrifugation method, and second is protamine aggregated method. Next is phase behavior. At transition temperature, these liposomes undergo reversible phase transition. So this transition temperature is the indication of stability, permeability, and also it indicates the region of drug entrapment, which is done by DSC, that is differential scanning calorimetry. Fifth is drug release rate. The rate of drug release from the liposomes can be determined by in vivo assays, which helps to predict the pharmacokinetics and bioavailability of the drug. However, in vivo studies are found to be more complete. Liposomes encapsulating the tracer insulin are employed for this study. So this insulin is preferred as it is released only in the ECF, and it undergoes rapid renal excretion of the phase tracers coupled to the degradation rate constant of the tracer released from the liposomes. Now next is application of liposomes. So these liposomes can be used as a drug or protein delivery vehicles. Liposomes you can use in antimicrobial, antifungal and antiviral therapy. Also, this can be used as a radiopharmaceutical and radiodiagnostic carriers. Liposomes can be used in cosmetics and dermatological studies. So with this, I want to conclude that these liposomes have been used in a broad range of pharmaceutical application. Liposomes with enhanced drug delivery to disease locations by ability of long circulation residence times and are now achieving all the clinical acceptance. Also, these liposomes promote targeting of particular disease cells within the diseased cell. Finally, these liposomal drugs exhibit reduced toxicity and it retains all the enhanced efficacy which compared with the free complements. Only time will tell which of the above applications and speculations will prove to be successful. However, based on the pharmaceutical applications and available products, we can say that these liposomes have definitely established their position in the modern delivery systems. So this is about the liposomes. If you have any question, you all can ask. Yes, everyone. Okay, then. Thank you.
Good afternoon, everyone. It's a great privilege and honor to propose the thanks of the vote of the thanks for the today's session. I extend my gratitude to our eminent speaker for Ms. Trupti Jadhav ma'am for her relevant address on liposomes, a novel drug delivery system, has focused on various points like what are liposomes, its basic structure, advantages and disadvantages of liposomes, method of liposomes preparation, the various methods like the pro-liposomes, freeze drying, French pressure cells, and etc. The evaluation of liposomes and the applications of the liposomes. My sincere thanks to our chairman, Mr. Albert D'Souza sir, for his constant encouragement, and thanks to Mrs. Elvina D'Souza ma'am, the Secretary of Elder Education Trust, all the other dignitaries of the management. A special thanks to our principal, Dr. Savita Tauro ma'am, for immense guidance and support. My deep sense of appreciation and thanks to all the participants who attended this webinar with a great willingness. The feedback link will be provided into your respective WhatsApp groups. Kindly fill it so that it will be helpful us to improve our program. Thank you all. Have a wonderful day ahead.